Hello students, today we are going to start a new chapter, that is molecular basis of inheritance. Molecular basis of inheritance means inheritance. We have discussed that there are certain things which are responsible for transferring the genetic material from parents to the offspring. Now we in this particular chapter we will be studying about their molecular behavior. What is the basically structure of it? How in a microscopic level or molecule level, what are the molecules or component it is made up of? So first we will be studying systematically. We will study what is molecular basis of inheritance. Means the characters which are transferring, basically what they are transferring. Are they transferring in a case of certain chemicals or some formulas? What are those? Let's see. First we will go for the definition of gene. As we all know, gene are transferred. Gene are responsible for a certain set of characters. So what is gene? It's a unit which performs three functions: recombination, mutation and self-reproduction. Recombination to combine. Means combine together, that is recombination. Mutation means certain sort of enhancement is being done to become something new, that is mutation. And reproduction means to produce themselves again, this is known as reproduction. This is a second theory or a definition of gene is a macromolecule, is a bigger molecule attached to undifferentiated protein thread. Various protein threads are there, so attached to it, that is chromonema, that can pass from one cell to another. Means it's a capable macromolecule tied up with the protein, and it is transferred from one cell to another and can transfer it from one generation to another. A cell can multiply, so it is transferable in case of cell and one generation or another. According to this definition, it's a macromolecule, it's a bigger molecule which is tied up with the protein. So that structure we basically know as a chromosome. What is chromosome? It's a genetic material, gene with protein tied up. That is a coiled structure. So this is the definition of gene. Okay. Now, as we all know, the gene are the basic hereditary unit. Now we will go inside that gene. What that gene is made up of? As we are going to study the molecular basis of inheritance, so we must be going inside it. What basically this gene is made up of in a chemical basis. So before going to this, we must be knowing certain properties of a genetic material. What genetic material do inside the body or inside the cell? First, it duplicates always. As gene is inside the cell, what it do? It duplicates so that it will help in replication. Once it's got replicated, then if new formation of new cell happens, so that cell also will be having the same information, hence can perform at the same level. This is the first thing. Second, it mutate. It changes to the adaptation and it becomes something new, advanced every now and then. So it may becomes more sustainable in case of inheritance. And last, it transcript. Transcript means to decode certain set of information with the help of mRNA. mRNA stands for messenger ribonucleic acid. Messenger RNA. Messenger RNA means it takes message. A certain gene, a messenger RNA read it and then it goes to the target site and transcript it. This process is done when the duplication of gene is there or transcription of genetic material is supposed to be there. Okay. Now, structure of DNA. First, DNA stands for deoxyribonucleic acid and RNA stands for ribonucleic acid. Okay. Chromosome are made up of nucleoprotein. Ribonucleic acid means nucleoprotein. This is the protein. Okay. Now, DNA, there are various structure given by various scientists regarding DNA, as it's a coiled double helical structure, most popular structure given by scientists Watson and Crick. So there they elaborated what it is made up of. Basically, DNA has a nitrogen bases. What are the nitrogen bases? They are purine and pyrimidines. If we go inside the purine and pyrimidines, we will find adenine, guanine, cytosine, and thymine. Before you get confused, let's go for the table. Now, this is a table. This will give you the clear picture of what DNA has. Now, in the first column, you will see DNA. DNA has base, nitrogen bases. What are those? First adenine, second guanine, third thymine, and fourth cytosine. Up now coming to RNA. RNA what has? RNA has all the similar things. Same adenine, same guanine, same cytosine, but in case of thymine it is uracil. Okay. Now let us study regarding the attachment. How these are attached. Always keep this in your mind. Adenine, it attached to the thymine, and guanine is attached to the cytosine. Okay. Formula D, there is sugar. DNA deoxyribonucleic acid होता है, तो D stands for the sugar molecule. So always sugar molecule attached to adenine. It will form deoxyadenosine. Whenever sugar, that is D, Na, like this na. So D sugar get attached to the adenine, तो होगा deoxyadenosine. If guanine, guanine. If thymine, thymidine, and if it is attached with the cytosine, then deoxycytidine. Same with the RNA. If RNA, that is ribose sugar. In DNA there is a deoxyribose sugar, and in RNA it's ribose sugar. So with adenine it's adenoside, guanine it's guanosine, uracil is uridine, and cytosine it's a cytidine. When nitrogen base attached to the sugar, it forms nucleoside. Keep this in your mind, because as you move further in this particular chapter, you must not get confused by this, कि what is nucleoside and what is nucleotide. Okay. So when sugar particle combines with nitrogen bases, it forms nucleoside. Then what is nucleotide? This is nucleotide. Nucleotide means in nucleoside one phosphate particle is attached to it. As you say before, below D, P is attached. P stands for phosphate. Okay. You can say like monophosphate. Okay. Single phosphate is there, so it can be monophosphate. So when phosphate get attached to the nucleoside, it form nucleotide. Okay, you can see in the figure. Understood? Now this strand nucleotide is a half part of the genetic material. When this half part will join to the other half part, then the DNA will be formed. Okay, let's see that those structure on a molecular basis. This is the molecular structure. Now I told you guanine is in this figure base pair. G is attached to C with a triple bond. I told you guanine attached to the cytosine with a triple bond. T attached to A with a double bond, double dotted lines. This stands for double bond. These dotted lines are representing the double bond or a triple bond. Okay. And a whole structure is sugar phosphate chain, and the middle bond is known as base pair. Now, if you see outside the figure, the direction is being given. In one side it is 3 prime to 5 prime, and another side is 5 prime to 3 prime. What is this prime? 5 dash, 3 dash in the corner you find. This is the position of pentose sugar. You can see guanine से there is S written. There is a 5 corner substance, sugar molecule, pentose sugar बोलते हैं उसको. क्या बोलते हैं? Pentose sugar. तो its fifth position remains empty, then that end is known as 5 prime. If third position is remain empty, that is known as 3 prime. So this is how half genetic material is joined with the help of bonds and forms a complete pair of genetic material. This you can see in this. Now this is a more zoomed chemical structure of base pairs and sugar phosphate. Now you can see the molecule of cytosine, how much complex it is. Guanine you can see, okay, adenine you can see, thymine you can see. So basically what we are studying before in a form of A, G, C, T, and we are having a genetic structure, that is DNA structure, very well structured double helical twisted structure, right, Watson and Crick's. Now this is how we are made up of chemicals. These are the chemicals, how much nitrogen is there, how much cytosine is there. Okay, inside below cytosine you will find the pentose sugar. 1, 2, 3, 4, 5 positions are given and 5 prime is empty. So if the 5 prime is empty, then that end will be 5 prime. I hope you are getting what I am explaining. Now come to next. Some physical properties of DNA. What are the properties of DNA? First, molecular weight 10 Dalton. Molecular configuration means 3-dimensional figure it is. It's 3-dimensional. Diameter 20 Armstrong. Number of bases in one turn 10. In one turn there are how many number of bases? 10. Length of hydrogen bond is 3 Armstrong. Diameter of a base pair in 2 strands is 11 Armstrong. Distance between 2 adjacent bases is 3.4 Armstrong. Distance between 2 complete turns is 34 Armstrong. Okay, now you have to remember these things by heart. This is the basic start of the chapter. Basically today this is all for the day, but for next class you must be reading this topic properly and preparing notes for it, and make sure okay, you will read it, because in the next class we are going to study the more detailed structure of DNA, how it is formed, transcription, a lot of things are there. Okay, so that's all for the day. Thank you very much.